The amount of land that's been dedicated for palm oil has increased exponentially in the last sort of 40 or 50 years. So there has been a lot of deforestation. And with deforestation, of course, there's loss of biodiversity. Humankind has got a big challenge. By 2050, the world's population is going to exceed 9 billion. To feed that number of people, we're going to have to double the amount of food that we're producing today. We have to close this food gap in the next 30 years, but we can't do that by clearing more land for agriculture. With that need to increase our productivity as much as we can, we really need to boost the yield of the individual palms that you see around. So that led us into many different projects on how we can get the most productivity we can from the land that we have. With that in mind, we looked to genomics as a way to do that. And it took about 7 years of sort of basic research that enables us to look and analyze the DNA of a palm and to predict how good the yield will be.
You can't separate science from innovation. 2 years ago, Sime Darby Plantation has embarked into a project called Genome Select. What this project is entitled to do is to screen for high-yielding planting materials for oil palm. We have used all the information that we know for the past 7 years of studies from the genome information to understand where are the differences lies in regards to high-yielding materials versus low-yielding materials.
Genome Select is about 15% higher yield than our previous best planting materials. Commercial yields for oil palm are around 4 to 6 tonnes of oil per hectare, but these palms can produce above 10 tonnes of oil per hectare. If we can realise those kinds of yields across all of our planting environments, we can close that yield gap by 2050. Once Sime Darby Plantation has the over 300,000 hectares of land that we have planted in Malaysia, planted with Genome Select, the amount of additional oil that we can produce is equivalent to 50,000 hectares of land that wouldn't need to be cleared. Oil palm has got a lot of challenges. It's not going to be easy. If we continue to be passionate about where we want to end up, I think we'll get there.
What is a genome? A genome essentially is the book of life. It contains all the information to make that oil palm tree. We're publishing the oil palm genome, and it's 80% more complete than the previous publicly available genome. By using the genomic information, we have actually demonstrated that yields can be dramatically increased. And we hope that by making this information available, the whole industry will benefit from this, and we can actually deliver additional oil without increasing the impact on the environment.
By 2050, there'll be around 10 billion people on this earth. That's going to result in around a 70% food gap over and above what we're producing today. That's going to require more hectares of additional agricultural land to produce that amount of food. That's huge. So the only way to solve this problem is increase the productivity, increase the yield of the land that we already have. That's going to require a step change in yield. It's going to require breakthrough innovation to be able to meet that food gap and put a stop to deforestation.
It's wonderful to actually manage such a great, young, enthusiastic team of people who work from the lab doing fundamental research all the way along until the field. So what we managed to do is connect lab research to the plantation. We've actually validated that the use of the genome could result in much higher yields and much better performance from our planting materials. We've actually planted our Genome Select palms, and in the first year of commercial yields, they have gone up by over 20%.
We had to develop a number of technologies along the way that enable us to basically track the seeds, ensure their quality all the way through to the field, and through GPS and satellite imagery now we can really monitor the performance on a very large scale of individual palms in a way that was never possible before.
What we're doing now is to scale up production of the Genome Select and plant it across larger areas. And we are trying to incorporate in new traits such as climate resilience, disease resistance, and also better harvestability.
One of the key things that the research and development team at Sime Darby have achieved is the whole integration of molecular genetics into the breeding program. It's the application. The application is not trivial. In fact, the application is probably 9/10 of the work from the fundamental science. All credit to Sime Darby research team. This is going to be a model for how the same approaches could be applied in practice to try to take things through to actually giving increased yield on the same land.
The program has sought the basic truth about the oil palm genome and what can we do with it. The technology has been scientifically sound and verified and reproduced. It has led to a more sustainable product at Sime Darby, which I think is a critical factor in this time. It's developed a formidable team of people at Sime Darby who can do very exciting, cutting-edge interdisciplinary research, which is competitive throughout the world, a fact that I'm very, very proud of and to be associated with.
Very good morning, very good afternoon to everyone, regardless of whether you are on this planet, and welcome to this Breakthrough Innovation webinar, which will be discussing about discovering the oil palm genome for sustainable growth. Back in November 2019, Sime Darby Plantation opened up the discussion on the critical role that innovation on yield will play in the sustainability of the palm oil industry. It was brought up on the premise that the rapid rate of deforestation is an urgent challenge for the world that demands a meaningful response. People are concerned that palm oil production is a contributor to deforestation. And as a leading producer of sustainable palm oil, we share their concern and are working towards a deforestation-free industry. In June last year, in 2020, Sime Darby Plantation announced the sharing of its oil palm genome research, which was eventually published in Plants, an international open access journal published by the Multidisciplinary Digital Publishing Institute, as well as made available on the website of the National Center for Biotechnology Information, or NCBI. While we had discussed why innovation to increase yield matters so much to our industry in a similar gathering held in July, we're here today to share our journey on palm genome research and plantation innovation as part of our mission for a deforestation-free industry. I'm so pleased that we have with us in this virtual gathering a mixed group of people, because it is a subject that is relevant to all of us, and it will take the entire industry and everyone involved in it to deliver innovation for the future. I'm very pleased to note that we have over 100 participants who are joining us online this afternoon or this morning to listen to what we have to say today. I'm joined here today by our Chief R&D Officer, Dr. Harikrishna Kulaveerasingam, and we have next to him Dr. David Ross Appleton, who heads our biotech and breeding team. At the far left, Mr. Tae-Chi King is the principal scientist at Sime Darby Plantation's Technology Center. And closest to me, I have Ms. Ling, the chief scientist for molecular breeding and bioinformatics. I'd also like to introduce to you a special guest who will contribute her perspective on the work that's been done to get to where we are today. We have with us online Dr. Rhian Gwilliam, Director of Field Applications and Technical Sales from LGC Biosearch Technologies UK, a valuable partner who has helped support Sime Darby Plantation's journey. We couldn't be more delighted to have you with us, Dr. Gwilliam, to share your thoughts on the work that we've been doing together. But before we start, before I hand over the session to Dr. Appleton, let me set the scene. First of all, let's recognize the context that there is a challenge in the world for this industry. In the coming years, there's expected to be a significant growth in demand for food and for oils from a growing population. And just meeting that demand will not be easy. And striving to do so represents the potential for significant growth for the industry. Yet, at the same time, we all face a growing climate challenge, and there is an increasing urgency to halt the continued rapid rate of deforestation that contributes to that. People all over the world are deeply concerned about the oil palm industry as a driver for this, but for Sime Darby Plantation, it's the same concern for us. And to rise to this challenge, we need to change how we as an industry operate today. The industry needs to innovate to increase yield, because that is how we will be able to produce more oil without further land clearance. Without further ado, I'm now pleased to introduce Dr. David Appleton, who will share with you our story on Genome Select, Sime Darby's high-yielding oil palm developed using genetic testing tools, as well as its future direction. Dr. Appleton, over to you.
Thank you, and good afternoon, everybody. So today I'm going to give you an update on Genome Select and some more about its future direction with us in Sime Darby next. So you heard from Alfian just now that deforestation is really a huge challenge for the world. How can we feed a growing population and put a stop to deforestation? We have to increase our efficiency. We have to increase the yield on the land that we already have. And in order to do that, we're going to need to be able to identify, optimize, and perhaps more importantly is scale technologies and breakthrough innovations and see them come to reality. And you'll see today, it's not always easy to do that. There are big challenges. But if we keep our eyes open during this journey and we keep looking out, we can learn from this and we can further improve. Next. Next. So Genome Select was a 12-year journey for us. Sorry, back. Genome Select was a 12-year journey for us from 2016, where we had our first planting of Genome Select palms, which you should be able to see in a minute. And even before that, back in 2008 and 2009, when we assembled the team to first sequence the oil palm genome. And through to today, when we're starting to see commercial yield of these palms and on track for producing 1.5 million Genome Select seeds this year. We won't stop there. We'll be planting seeds in Indonesia and also in Papua New Guinea. In fact, we've already rolled this technology out since 2019 to give a premium seed offering to our customers that buy seeds from us, where we can give them genetically authentic seeds that are traceable right through to their fields. That's called Calyx Q6. Next. But let's not forget, our challenge was actually to double yield by 2050. This is something that the world needs. It's something that has to go beyond Sime Darby Plantation. The whole industry has to rise to this challenge of doubling yields. As I mentioned, it's a big challenge, but we can really see that this is possible. Next. So when we planted Genome Select in 2016, we now started to see commercial yields. And the, sorry, back one slide. Oh, okay, sorry. So early yields before we started going commercial were very encouraging from Genome Select. Next. But once we started to see commercial yields at 3 years old, we started to see a difference between the Genome Select palms planted in two different locations, one being coastal, which is the kind of optimum conditions for oil palm, and the other one in inland. Both of these locations, the yields were superior to surrounding fields that were planted at the same time. But the difference in the inland soil in the red line there was much less than what we saw for the coastal, which was really very impressive. But worse than that, I think we saw about 4 months where there wasn't very much yield at all from the inland planting of Genome Select seeds. This is actually quite a common thing. And it's due to stress. It's due to a difference in weather conditions, water deficit or drought, basically, in the inland area. Next. But with the help of digital technologies, we can now track this. This is an aerial view of that field. And the little points on this field that you can see are the palms, which at certain times in August and February, and we have a very regular mapping of this, aren't producing any bunches at all. So we can see a big change between the seasons. So what we can do now is we can track that, and every single palm, all 9,000 in this field, have a genetic fingerprint that we have. So we can start to analyze this. And this is giving us insights into how palms respond to stress and water deficit, and how these fluctuations in seasonal yield occur. So we can use that now. We have a high-tech nursery where we can test this out under controlled conditions and really investigate how palms respond to this stress, and take it further so that in the coming years we'll be able to have high-yielding and drought-tolerant or climate-resilient palms planted in our fields for Genome Select. Ganoderma is also a big challenge for the industry and something that's getting worse. It's a particular fungus which affects the palms. And we've been able to apply the same technology to look at how palms can defend themselves against Ganoderma. And using Genome Select, we can now have Genome Select models that can select for Ganoderma tolerance. In fact, this year we're going to move those into the field to validate them. We've put them through the same high-tech nursery that I mentioned for the drought tolerance. And these will be tested this year in field planting. Next. So on Sime Darby Plantation, we're very fortunate that we have breeding locations and seed production centers globally around the world. So in Malaysia, we now have more than 3,000 hectares of Genome Select planted in 19 different estates across 6 states in Malaysia. We have seeds that are tracked all the way through the field from the parents, from the parental palm that produced them, and we have very detailed yield analysis. We now have mother palms of Genome Select in Indonesia so that we can produce Genome Select from 2024 in Indonesia. And in Dami in Papua New Guinea, we are also applying Genome Select to the breeding materials that we have there. So in the coming 10 years, we'll be able to test alongside each other these different materials in the different locations to see their performance and head to having basically optimum yield in all different locations that are relevant to oil palm around the world. Next. So I just wanted to reiterate, in order for us to realize that massive step change in yield that we need to have breakthrough innovation. But having high yield in one test plot or one trial plot is not enough. In fact, having high yield in one commercial location is also not enough. We have to have high yield in all locations at all times. And this is what we're heading towards. And we're putting the various technologies in place to make sure that we can realize that in the coming years to come. And that's how we are going to realize a doubling of yield by 2050. Of course, we can't stop there as well. We need to address those other really critical issues for the oil palm industry, such as labor around harvesting and these kind of difficulties. So as a company, Sime Darby Plantation, we are dedicated to addressing all of these challenges and putting it all together to make sure that we can feed the growing population without any deforestation. Thank you.
Thank you very much, Dr. Appleton. Right, ladies and gentlemen, moving on to share with you on our journey to get to where we are today. I call upon Mr. Tay Chi King, the Principal Scientist at Sime Darby Plantation's Technology Center, to share his presentation. Chi King?
All right. Welcome, everyone. A very thank you for everyone to attend the sessions. So I would like to share some of the journey that Sime Darby Plantation have went through. So we actually have started this genomic project actually about 10 years ago when we have sequenced the oil palm sequence. So I noticed that there are so many audience coming from different areas. So please allow me to introduce what is actually genomes. So genome actually is not a fancy thing. Actually, for every organism, including humans, we do have a genome in our cells. So in the genome, it's a place which consists of all the genetics and the genes that actually have the impact on all the biological functions and also the morphologies of one organism. So for instance, in human genomes, we know that there's a 46 piece of chromosomes and everyone put in pairs because in science, we call this is a diploid genome. And 50% of them is actually inherited from our father, and the other half is from our mother. So when we try to unveil the chromosome, you'll find that the famous helix, double helix DNA structures is actually sitting in the chromosome. And there is 4 different bases in sequence in making everyone looks the difference. Next, please. So I believe that most of us have heard from the newspaper or news saying that the first human genome has been successfully sequenced in about 20 years before. And now we are not limiting in 1 human genomes. Actually, we are moving to 1 million human genomes. So one of the objectives here is to customize the medical treatments to every single individuals because all of us, they are all different. So now in animals and also the crop research, we are trying to follow the same strategy. So that's why every year you have about hundreds of the new organisms being sequenced and published in the journals. So actually, when you receive the data, you will see on the slide here is actually you have a whole, maybe a few books of the ACGT in sequence, okay, without any clue. So that's why we have, as a geneticist, actually we really need to study and locate which portion of the genome actually have the functions and influence to the trait that we are interested in. Next, please. All right, so we put use oil palm as examples. The whole page of the codes is actually served as an oil palm reference genome. So we will put at number 1 of the slide there, and then we try to superimpose with the different individuals that we are interested in. So you notice that some portion throughout the genomes, they actually different. And the differences here is actually known as a genetic variation, and this mainly due to the mutation happened during the evolution process. So interestingly, not all the differences here is actually having the impact on the traits that we are interested in. So for example, in oil palm, we have 2 major groups. One is a simple traits. Okay, we have the shell thickness, dura, piscifera, and tenera, and also the fruit color, the virescence. So this type of traits is very simple. It's actually controlled by 1 or 2 genes and follow the Mendelian inheritance. So we have our MPOB colleagues actually have published and find the causal genes for these particular traits. And now this is where the DNA markers are available to predict that type of traits. But for the other economic important traits, just like the P&D, pests and disease like ganoderma infections, the trunk height, the oil quality, and also the oil yield especially, they are extremely complex in nature. So meaning to say they are actually controlled by not only 1 or 2 genes, they are actually many. And because of that, that's why we need to spend more effort to identify all these genes throughout the genome. So the map here is very important. And at the same time, as mentioned by Dr. Appleton, this type of trait is very susceptible to the environment. So that's why we have to address both together. Next, please. So this is where the journey started in Sime Darby Plantation. So we started, like the other research institute, with the linkage mapping approach. So basically, every single line there is just a piece of DNA as the markers, and we locate them in the 16 chromosome groups based on 1 or 2 specific families, the recombination rates. So by using that one, actually, we managed to identify some of the important QTL regions for the certain like oil traits. But the reason, because they are too sparse, that's why we only can apply this type of marker in the certain families only. But in 2013, there was the first oil palm physical genome known as P5 built version published by our MPOB colleagues in Nature paper. Because of that, this opened a new way for the genome-wide association studies by putting all the 200,000 SNPs on these 16 chromosomes together. So as you can see that from the first version until the GWASs, the Manhattan plot there, you can see how much marker density has been improved. And this is where we have found the 3 major QTL for the mesocarp oil contents. So traditionally, we will try to use that 3 major QTL for the marker-assisted selection, but because of the cost of the genotyping is going down, so that's why we decided to include all the markers that we have in the yellow box and put in the genomic selection approach. So classical genomic selection is actually, they will not require the pre-knowledge on the QTL. But what we found that actually, if you have the QTL information plus together with the genomic selection, the prediction accuracy is significantly higher. So that's why in 2017, we have published this particular research. Next one, please. So after having the model, of course, we would like to immediately apply in our commercial field. So we decided to apply the model to predict the yield prediction of every single seeds, namely Calyx 600, okay? Because in this population, there is a variation in terms of the yield. By selecting the top 30%, we can immediately have 16% oil yield within the same generations. So as you can see, this technology actually, we don't actually modifying the genomes or any DNA, and they are not clones. So they are 100% like the normal D by P seeds. So one key success to enable the commercial deployment is we need to have very robust and high throughput genotyping system. This is why we are very happy to have a strong partnership with LGC. And the system here is actually built in our Sime Darby, and it used to be the largest lab in Southeast Asia. So after having the facility, this is where from 2016, we managed to roll out the whole Genome Select program. But we never stop our research here. So along the time, we are trying to optimizing the marker density, because once we can reduce the markers, then the cost will go down without compromising the accuracy. Secondly, we actually have moved out from the screening for every single seeds, but we are actually profiling the parents, and we try to simulate what is the combining ability between the father and the mother. From there, we know which couples actually can produce the high-yielding genome select materials. And by doing that, actually, we managed to cut down our cost, like 99% of our testing cost. And at the same time, after planting them, we are still doing the field monitoring and validation as mentioned by Dr. Appleton. Okay. Lastly is, because we are planted everywhere on different environments, this actually gives us opportunity to study the G by E interactions here. Next, please. So this is the strategy here. So within the commercial planting, on the right-hand the box there, we actually have set up the RCBD design with the proper negative controls. So we have the different ranking of the yield based on the genomic selection model we have, and then it's surrounded by the normal commercial planting. So within the one box, actually, after having all the data, we managed to validate our GS prediction model. By putting more than one location, we have Sabah, Sarawak, we actually have our sister company like MPPO in Papua New Guinea, and also Indonesia, Myanmar. We are now able to identify whether the response, the yield to the different MI, they are parallel or they are contradict to each other. If they are contradict, meaning to say we will require a new prediction model to further improve our prediction abilities. So far, we have planted 3,380 hectares since 2016 across these regions. Okay, next. So this is the highlight of the premium seed development in Sime Darby plantations. So in 2008, actually, we have sequenced the first oil palm genome. Okay, 2010, we actually have branded Calyx 600. This one, 100% is based on the conventional breeding programs. And 2013, we have our MPOP colleagues actually published the oil palm genome, the first physical map here. So they enable all kinds of association study after that. And we also have working together with Orion Bioscience so that we are using the shell marker and also the virusen marker in our breeding program, especially in the pollen development program. In 2016, we managed to launch the first genome select materials. So from 2016 to 2020, the total hectareage we have covered is 3.4 thousand hectares. So in 2019, the Calyx Q6 is actually an improvement from the Calyx 600 by ensuring the genetic purity of the material they have produced. And in the same year, we managed to improve the oil palm genome assembly from the P5 version to PMV6, which will be explained by my colleague Ling later. So our ultimate aim is from 2023 onwards, we would like to plant genome select material to supply the full Sime Darby plantation replanting, which is 5% per year. So with that, thank you very much.
Thank you very much, Chiking.
Yeah.
Right, as Chiking mentioned just now, to follow up to his story, we have next with us Ms. Ong Ling, who's the Chief Scientist for Molecular Breeding and Bioinformatics. So Ling will be sharing with us her experience in putting together the genome sequencing that will help improve the palm oil industry and other palm members. Ling, over to you.
Hi, good day, everyone. So glad to be here to share about the recent improvement of the oil palm genome assembly as well as its application. So if we were to use the analogy of genome assembly between genome assembly as well as a jigsaw puzzle, it all started with small pieces of the jigsaws puzzles, and then we normally the logical way we will group all the puzzle into similar patterns into small islands, and then there's normally no problem to build them in a locally manner in a perfect way. But however, after that, if we want to join the small pieces puzzle into a global view, it comes problem, because if we don't have a picture to be referred, we don't know where to join all these kind of puzzles. So the third picture at the right corner seems like a complete one. In the realm of a jigsaw puzzle, it might still not complete, but for genome assembly, it seems to be a very good picture, because we can now see the whole picture. What is the picture telling us? Is a scenic view of a city. Despite the small missing pieces, it's just a minor problem. So next. So for our journey in genome assembly improvement is all start from the linkage map built from SSR-based markers as shared by a colleague just now. So we can see that generally the markers are sitting in quite sparse distance. We then evolve into SNP-based linkage map where we significantly narrow down the distance between the markers. However, this approach is only using single mapping population. We then thought of maybe we should increase the mapping density and move on to even more population, and then consolidate all the population into one in order to maximize the marker mappabilities into the linkage map. So in 2019 and 2020, we consolidate 3 mapping population. They are actually the 3 of the largest mapping population in our breeding stock, which with the family size of the progeny ranging from 100 individual to 300 individuals. So that's maximize the density. From there, we start to examine the relation of the linkage map with the genome map as published by MPOB. So the right corner, bottom corner is actually the improvement that we have made from the published version of the genome. Our way of making it is actually breaking the original assembly into small pieces of the jigsaw puzzle again, and filling up all the different previously unplaced genomic pieces and extend the genome. Next. So how do we ensure that our assembly is the right one, or the ordering of a marker is the correct one? So we first evaluate the relationship between our linkage location of the marker and compare against the genome map from P5 build version, which is the 2013 one. We found that they are actually very highly correlated, which is close to 1, which means that from there we get the confidence that our ordering of marker is correct. And that's why I would like to bring attention to, for example, population 3, the initial way we can't see the good relationship, and that's why we increase the mapping resolution into 3 mapping population. So from there we have the we move on to improve the genome version by anchoring more of the genomic scaffolds into the genome. This is an example from 1 chromosome. So we have originally extend from 40 MB to now close to 80 MB. And we examine the relationship between the genetic location as well as the physical location of markers. It appeared to be what expected, which is a sigmoid curve. Next. So the same approach has been repeated over the 16 chromosome, and this is the statistics of the improvement. In terms of length of the MB, we are now able to actually extend most of the chromosome except for the minor chromosome, which is a chromosome 16. And in terms of number of scaffold that can be placed into the chromosome, we have also putting about 80% of the scaffold can now be assembled into the genome map. So each of the chromosome now has averagely 73 MB compared to the P5 build, which is 40 MB. And the number of scaffolds now can be found in each of the chromosome is about 140 compared to previously about 20 pieces of genomic scaffolds. So the right bottom corner is again the comparison between the map of the P5 build and the PMV6, which is our latest version. We can see that now the resolution has been increased and fill up a lot of gaps. So this has immediately helped us to enhance the mapping resolution for our genome-wide association studies, as well as we are able to find or locate the genes into different chromosomes. Next. So take a glance at our first round of reported genome-wide association studies. At the top is the P5 built genome originally. We are able to find the major QTL as reported by Chiking just now, but we found a chunk of unmapped SNPs initially. So we just put them into a chromosome 17 or just unmapped SNPs that we cannot locate them within the chromosome, and thus we cannot know where is the QTL for these parts of the SNPs. So when we have improved and relocate all the markers, so we are now able to see the sharp peak at the major QTL at chromosome 5, and the number of unmet SNPs are significantly reduced. So if we were to zoom into the chromosome 5 as an example, initially the peak, it seems to have two peaks. It's like we don't know where to focus because there's two peaks, but after we have relocated everything and remap, then we are now able to see there's one peak that is easier for us to hunt for the causal genes. And again, to mention that the length of the chromosome has now been increased from 40 to 80 MB. So the next thing is how this improved genome again will help us. As I mentioned, we have placed additional about 20%, 24% of the genes into their respective chromosome. It is for the hunting of the causal genes that is reside within our QTL region. So take the bottom left as an example. The improvement of genome allow us to visualize it in a genome browser, where we can now locate all the markers, the variant, the genes, and the transcript that tells us where is the location of this kind of information. And what's more interestingly is we are able to observe the linkage disequilibrium between different populations. So it is very important for us to know that how actually the selection pressure works on different origins, and why sometimes certain marker work in one population but not in another. So the right corner is actually example of how can we further manipulate gene of interest when we have a better coverage of a gene of interest. So what's next? If we have a better coverage of the genome, we can move on to perform comparative genomic studies between palms. Take this example, we use coconut, date palm as an example, as a close relative of oil palm. And then we are attempting the same way to place all the scaffolds using oil palm as a reference in order to rebuild the date palm genome. We thought of why don't we try and using this same approach, whether we can improve the date palm genome or not. So at the bottom left corner, there's a correlation graph that shows that the x-axis is the original oil palm directed genome assembly improvement of the date palm, and the y-axis is the recent date palm assembly release. And we can see that actually what we have used as oil palm directed genome assembly can somehow correct the date palm, can somehow assemble the date palm genome in a correct way. Next. So as a conclusion, with the current genome that we have, we are able to unravel some of the interesting and important agronomic traits that we have now, but there's still room of improvement in the genome. So we are urging all the industry or the academic layer to work together to build a better oil palm genome map. That's all. Thank you and good day.
Thank you very much, Ling. Ladies and gentlemen, before we go on to the next speaker, I would just like to remind the participants who are joining us online that you can start to type in your questions in the Q&A section of the app so that we can either read it out or we can call on you when you raise your hands during the Q&A session right after this. All right, without further ado, as I mentioned earlier, Sime Darby Plantation looks forward to mutually beneficial partnerships with like-minded organizations. And in our journey towards discovering the oil palm genome, we work closely with LGC Biosearch Technologies of the UK. And I'd like now to call upon Dr. Rhian Gwilliam, the Director of Field Applications and Technical Sales from LGC Biosearch, to share the work that we've been doing together. Over to you, Dr. Gwilliam.
Thank you very much. I'm delighted to be speaking today. So my presentation today is going to be telling you all about LGC's journey with Sime Darby in partnership to accelerate the genomic breakthroughs that they've been working with or towards in palm oil. Next, please. So LGC has a really wide remit. We have lots of sites in 34 operational sites worldwide. And from our perspective, we are looking for partners to assist that we can share our technologies with in support of their academic endeavours. Next, please. So LGC has 4 different divisions, one covering clinical and molecular diagnostics, one working with pharma and biotech, one in the applied sector where Sime Darby sits, and one helping research and government. We have lots and lots of different tools that are provided to give solutions to the scientific issues that are present in everyday research. Next, please. So with respects to Sime Darby, we are looking at food and consumer products, and we are certainly focused on helping seed producers and animal breeders, looking at things such as feed conversion efficiency, sustainable practices to minimize resource use and impact environmental or for environmental benefit. We also work with farmers and food producers, regulators and food safety testing labs, and retailers and consumers. So ultimately our aim is to cover the whole of these sectors for the benefit of science in a safer world. Next, please. So what was the reason for the partnership between LGC and Sime Darby? What is the issue that Sime Darby needed to tackle? Well, we know they had 80 million data points or 80 million SNPs that they needed to be, let's start again, 80 million samples that they needed to screen in 6 months duration. And from our side, we have the tools to help them. We cover everything from SNP discovery through to QC. So where Sime Darby has been sitting is in that marker assisted selection. And from our side, we fit nicely as a potential partner here. We provide the tools and we provide the opportunity to design assays for those markers, to give you the information, and then we give you the automation to cover that. Next, please. So from our point of view, we have the discovery, GBS and NGS services from LGC and the whole genome sequencing with our library prep kits. And from our side, where Sime Darby required to use us was looking at that service aspect to develop the markers, to test them, and then to look across to see which of those markers would be informative for the testing. Our SeqSNP offering gives us the opportunity to look at higher density of SNPs covering that sector. Next, please. And then we have the PCR, qPCR, and multiplex option, where our CASP chemistry, and that's the driver for us, is to use our CASP chemistry to provide the opportunity for you to run these markers within your own labs. Next, please. At the preface of this, we actually look to see how can we allow you to take the leaf from the field and to extract the DNA to enable you to do the testing. And our SpeedX chemistry has been well used in Sime Darby's hands to extract from leaves and from the palm oil itself. Next, please. So we offer solution services for your challenges. We have the extraction, so we will take your leaves and seeds and extract either DNA or RNA. We will look after your samples, and then you can decide what you wish us to help with. So we like to say we can offer you sequencing, so either the Sanger sequencing, next generation sequencing, or targeted sequencing, plus the bioinformatics support after that sequencing process. Or we can help with the genotyping. We can support you in using our CASP genotyping, our all-inclusive service where we do the extraction and the genotyping for you. We look after you by doing the assay design and validation, and then we consult with you on the data analysis. So we're a complete integrated service provider. Next, please. So the challenge from Sime Darby back in 2015 was to set up a high throughput genotyping laboratory, the first in Asia. And currently there was no genotyping performed at Sime Darby. Develop the protocol for the automated extraction using SpeedX and our Octopure automation. Develop and test those CASP assays for the oil palm genotyping. So we actually designed and tested them in our own labs before transporting them across to Sime Darby for their use. And then look at those 80 million data points within that 6 months. Next, please. So this is just an overview of our own lab. Our LGC service laboratory is the largest commercial PCR genotyping lab in the world. And we did that, performed that process for the initial 80 million data points in our labs in London in the UK. Next, please. Simultaneously, in the Sime Darby lab, we installed the equipment and we trained the Sime Darby team in the use of that equipment. As part of our partnership commitment, Paul Scally was exported from the UK into Malaysia, and he supported the customer on site for 6 months. So this is a picture of Paul getting the full Sime Darby experience, helping in the field as well as helping in the lab. And we have Dana Chow, who's our field application specialist that we have placed in Malaysia to support Sime Darby. So we are committed to supporting you long term. So how did we do this? Well, the high throughput genotyping platforms that we offer are the IntelliCube, which is a qPCR and a PCR instrument, the SNP line for 384 well plates, so lower throughput SNP line, our SNP line for 1536, which is the option that Sime Darby chose to progress with, and then we have a Nexar high suite for higher throughput for future proofing and for growth for companies. So Sime Darby decided on their requirement was for optimization, for low volume usage of reagents, but for maximizing sample throughput. So they chose to work with us to put the 1536 SNP line in their lab. Next slide, please. So here shows you the Sime Darby lab in action with the equipment. Next, please. And the next. So this shows you a combination of Dr. Sagana standing in front of the Octopure, with Amaron standing in front of the meridian. So it shows that they have daily use and high throughput use. So very well, a great team there at Sime Darby in being open to adopting these new platforms and new ways of working. It's a really great experience for LGC to work with a team in Sime Darby. Next, please. And the kind words of Dr. Ross Appleton, that the partnership with LGC was a success for both the in-sourced and outsourced elements of the partnership. So from LGC's perspective, this was really valued. But what was the impact of the partnership? How did this impact Sime Darby and the work that it does? Well, we've heard from previous speakers that there is a great increase in yield, 20% increase in yield using the Genome Select palms that they are now planting. Next, please. So from LGC, we offered the platforms, reagents and services for the validation and screening. We supported the reproducible data in the high demand situation. It allowed for that quick ramp up time for new sites, backing up the solution and the peaks of demand. And we met the required capacity. And hopefully we feel we sometimes exceeded the expectation. Next, please. We feel at Sime Darby that a great partnership goes a long way. And we're grateful for the team at Sime Darby for adopting the platform and working with us. And we are reaping the benefit of the great work that you're doing. And we have pleasure in inviting you to be speakers at the events that we are organising from the LGC side. Next, please. And it only remains for me to once again reiterate thank you to the Sime Darby team for working with LGC in that great partnership. And we look forward to working with you in the future.
Thank you very much, Dr. Gwilliam. I hope you could stay on as we will move on to the Q&A session very shortly. Right, ladies and gentlemen, we have much to think about after hearing from our speakers, and now is the chance for us to hear from everyone participating on this session. The more than 200 of you who are out there joining us online, where do you think the greatest priorities or opportunities are for genome innovation to move forward from here? So before we open the discussion, I'd like to kindly request everyone to contribute by either raising your hand so we can call on you, or putting a note in that chat box on your screen so you can read out your questions to the panelists. Right, we have the first question that's come from Indonesia, from Mr. Rizky Anjal Puji Nugroho. He says, I'm enthusiastic on oil palm breeding. To Dr. David, thank you for your presentation. I'd like to ask a question about how important is it about developing breeding population through any mating design in genome selection milestones? Okay. Again, the question is, how important is it on developing breeding population through any mating design in genome selection milestone?
Okay. Well, genome select and genetic technologies can only enhance what we have already. So breeding programs with proper design, different populations, and bringing these together to advance what we have further is something that's going to be critical to bring other traits into it and to push oil palm a step change. So genome select is really a tool to help us to be more efficient and faster in that process.
Right, our next question from Dini Astika Sari. Thank you for your talk today. It's great for us to know that Sime Darby got the very high coverage of oil palm genome assembly compared to the previous one. This is foremostly awaited for oil palm breeders so we can go forward to do precise breeding. We'd like to know if Sime Darby is about to start with genome editing using CRISPR or Cas9 or CPF1 module. Please excuse my non-scientific pronunciation, if any. She wants to know whether we're using these modules to accelerate the breeding process.
Okay, from a research perspective, we do look into these technologies. It's something that is a global discussion around improving agriculture. So yeah, we are researching into this. Yep.
All right, next question, locally. What's the difference between genome versus GMO?
Okay, I can answer this.
Please, Chiki.
Yeah. So can you hear?
Yes.
Okay. Genome Select basically is just to develop tools, and then we try to quantify the natural genetic variations. From there, we put the DNA profile in our model so that we know that what is the future yield for from this particular seeds or seedlings. So meaning to say we are actually not modifying or not changing the DNA or the genetic information in that particular plants. So, but for the GMO, is actually you're modifying, okay? You're changing the sequence. But for the Genome Select material, no. We are actually trying to predict which particular seeds or the father and the mother can produce us the high yielding materials. That's all. Yeah.
All right. Next question. How is Sime Darby Plantation's first genome planting performing? What are their yields? Is it better than the normal seeds as expected?
Yep, so far the yield is very encouraging. It's higher than we had expected in comparison to control fields nearby. So we continue to be encouraged by that. We're continuing to scale up our planting. As I said, 1.5 million seeds this year ready for planting. As I show in my slides earlier, the performance of Genome Select in some locations in inland wasn't as good as coastal. It's still better than non-Genome Select nearby, but this gives us a great opportunity to study this further and to make sure that in future we have Genome Select which performs well under all conditions. So that's really the focus of our research and commercialization pathway now.
Right, the next question is a two-pronged question. What is SDP's plan with Genome? Is it looking at planting 100% genome? Should we plant a mix of seeds, i.e. GH, Chamara, Calyx Q6, etc., rather than just one type of seed? In case something goes wrong, such as sudden attack or disease, how are we going to manage this?
Yeah, we will be heading towards application of Genome Select technology as 100% of our seed production. But it's worth noting this is not 100% of seeds coming from a single parent. It's a pool of parents where the Genome Select tool has been applied to select the best of them. Breeding will continue. Breeding will continue to bring new genotypes and new genetic variation into our planting materials. These are not clones, so there is still natural variation. So it's really just a tool to make sure that we plant the very best of what we have.
Right. Next question. So far as we understand, you select seedlings based on the marker of oil to dry mesocarp content. Following of field planting of Genome Select palms, would you please share the performance of oil content of Genome Select palms? How is the increase of oil content compared to those non-selected seedlings?
Okay, so far for Genome Select in the two locations that were planted in 2016, the oil content is about 3% higher than the non-selected palms nearby in that same location. So that's what we're seeing for oil content. We also see increase in yield, so that's the fresh crop yield as well.
It seems the crowd online is getting warmed up, so more questions are coming in. The next question is, when we apply the selection of 5% seedlings selected by marker, we will get more narrow genetic planted palm in the field. What happens about the risk of those palms facing the climate as well as pest and disease challenges?
Want to take that one?
Yeah, sorry.
I'll repeat the question. When we apply the selection, which is 5% seedlings selected by marker, we will get more narrow genetic planted palm in the field. How do we manage the risk of those palms facing the climate as well as PND?
Okay. Of course, I think in any breeding program, it's actually narrowing the genetic information. It's just a matter of time, right? So that's why along the process, actually, we will try to inject the new blood, for example, the other population or other origins, and then we keep on updating our prediction model. So that is a strategy. So I think we are trying to mimic the same conventional breeding program, like to narrow to the certain genetic parts that we hope we just like making it faster. So that's why we need to keep on injecting the new blood to our training populations. So that is actually we are in our mind always. Yeah.
Thank you, Jikin. Dr. Hari, this may be a question for you. What is your cost of GS compared to commercial breeding? Conventional breeding, sorry. Cost of GS compared to conventional breeding.
Cost of GS. Of course, research cost is quite high. But if you talk about the commercial rollout costs, first generation because of research, most probably each plant is about 1,000 ringgit a plant. Okay. The subsequent next generation material that we produce now being planted, the 3,300 hectares or 3,200 over hectares that have been planted. So those materials are going for a few cents extra from our internal costs per plant. Of course, it doesn't include our depreciated research costs, right? We're just talking about total cost of doing the test, right?
Right, we have the next question from Yang Ping Lee. How about Sime Darby's stand in planting oil palm clonal materials in commercial estates?
Okay, we don't have a wide-scale cloning program in Sime Darby. At the present, we do have a tissue culture facility which we are using to augment our breeding program. At the moment, we see scaling of genome select as something which is more scalable, enable us to improve our yields faster and more efficiently in the fields, and give that genetic variation that many of the questionnaires have been asking. Yep.
All right, we've got a question from Ms. Siti Dalila. She asked, what about comparison with historical yield data from previous planted materials in the same field? Is there such comparison made?
You want to take it?
Yeah.
Question again, comparison with historical yield data from previous planted materials in the same field. Is there any comparison made when we plant new materials with previous in the same field?
Yeah, I think we are still monitoring, but by right, we should compare at the same time with the control. If let's say we try to compare with the different time, weather pattern they are changing. So I think that kind of comparison is, yeah, it can be a reference, but it's not a right comparison, I can say. Yeah, we would like to have everything put in the same time, same management, same environment.
Yeah, I think I can add to that, you know, over a number of years, we would expect the 100 hectares that we planted in 2016, and more importantly, is the 1,000 hectares per year that we're planting after that, we would be looking to see multi-year averages of that increase to the point that these are the highest yields that we've had in the company. I think to compare the yield of one particular field from the previous generation of planting, so much has changed since then, from how the field is laid out, how the replanting's taken place, the density. So we haven't really been making that comparison. What we are comparing is the yield of those fields across all the other fields planted in Sime Darby Plantation at the same time. And even more importantly is controlled monitoring and comparisons in that same location. So we're continuing to do that. We continue to be impressed by that. As I said, 1.5 million seeds this year, and scaling to 100% replant is done on the back of the encouragement that we see in the yields.
Thank you, David. We've got from our online participant, Martin Rushworth, who's asking, what success have you had in identifying genetic markers for Ganoderma, and will you be able to develop material to reduce Ganoderma incidence? And the second part to that question, and would South American olifera genotypes be able to contribute?
Yeah. Okay, for the American type olifera, definitely there's many interesting traits, like for example, the oil quality and certain disease, right? But the almost always people concerned about the oil when we try to cross with the African type, the commercial types. So I think definitely the marker can be developed using the same strategy, because the integration, I mean, try to restore back the oil definitely will take very long time through the several generation, putting everything on the field and test one by one. So definitely the strategy can be used. But for the Ganoderma, I think the team, now we are trying to develop the population and evaluating that. And actually we are in the progress of working on that. Yeah.
Right, next question from Ze Ying Seng. Will there be any scientific publication available on the yield performance of the SD using Genome Select?
Yeah, absolutely. We'll be publishing that. You know, typically in breeding, we're looking to having 4 years of yield data before you can say that you have statistically significant yield results. So we will not publish anything before we have really statistically sound analysis of this done. Okay.
Right, from Chandra Utomo, how many applied probes for your genome selection?
Sorry?
How many applied probes?
How many
applied
snips
probes?
How many snips do you use?
Okay, so yeah, we started, you know, with a pool of just under 200,000 probes or SNPs coming from the several million which is in the oil palm genome. And after several iterations of identifying the ones that are linked to yield and the ones where we can design a high throughput testing platform around, as Dr. Gwilliam had mentioned before, we had basically a genome select model built on 1,000 of those high quality reproducible SNPs associated with yield.
Right, next up, Mr. Yasir Anwar thanks us for the information that's come from the webinar, and he's wondering whether, again about Ganoderma, for Genome Select, is Ganoderma Genome Select using the same KASP genotyping platform?
Yep, so we have, as I said, we have 200,000 SNPs which we're able to do testing, and by analyzing those SNPs, we can identify those which are related to Ganoderma tolerance, and then develop a very similar assay to roll it out using the CASP system. And as I mentioned before, we now have that model. We've done selection this year. They'll be planted in the field in high Ganoderma prevalent areas so that we can validate what we've seen so far in the nursery.
Maybe can I add?
Please.
Okay. So Ganoderma is a complex pathogen, and it's difficult to say that you have tolerance because our tests are in nursery seedling materials together with one very virulent Ganoderma strain. It doesn't mean that if you plant it outside in the field, it will be tolerant to everything, right? So in that context, in the test system that we've used, it shows some form of tolerance, right?
Thank you, Dr. Ari, for adding to that. From Nurul Iza Abghani, who's asking us, in your breeding program, how do you take into account any possible inbreeding effects? Inbreeding effects, if any.
You want to take it, Chigi?
Yeah. So the inbreeding process, okay, there's a few ways of doing the inbreeding. Try to avoid that. Okay, one is we actually do use the molecular markers to quantify whatever, we try to monitor from one generation to next generation, how much homozygosity actually can achieve. Actually, we have that analysis. So once there's certain levels, then we think that they also portrayed some abnormalities. Then I think that it's time to inject the new blood. So that's why, as mentioned earlier, we always remember that we need to inject the new blood for after a few generations so that we can enrich or broaden the genetic base. Then from there, we update our GS prediction model. So that is the way we are trying to take. Yeah.
All right, again, another question on a certain specific trait is, how is GS resilient to extreme weather, i.e. El Nino or even La Nina?
I think as seen in my presentation earlier, just like other palms that we have planted in areas where there's sensitivity to water deficit, areas where there's a very low water table, and we've had periods of quite long drought in Malaysia in recent years. We can see a very clear seasonal effect of yield, even in Genome Select. As I said, similar to the rest of the palms in that field. The fortunate thing is having 8,000 or 9,000 palms in a field, which all have a genetic fingerprint, and now we're able to map this using digital tools. We can actually look at the genetics behind that response to drought, climate change, and build genetic models that help us to select for that in future so that we can guide our breeding programs towards palms that are more tolerant towards it. As I said, doubling yield by 2050 seems like a long time away, but that's exactly what we need to be able to do to get us to that point.
Right, back to the Ganoderma issue again. Will the new planting materials be able to tackle the issue of Ganoderma and more importantly improve plantation husbandry through agronomic approach?
Okay, yeah, as Dr. Hari mentioned, Ganoderma is complicated. One of the benefits we have is breeding trials, which have been around for many years, and the parental pools where Genome Select was developed on is very old. Those palms have been in a very high Ganoderma tolerance or Ganoderma prevalent area. And those palms that we've selected are ones which are proven to have, or at least stand in the field despite the onslaught of Ganoderma over almost 30 years now. And then using those to develop genetic models to predict tolerance in a nursery setting, this is what we hope we can now start to validate in the field. So yeah, what was the last part of the question?
Now, whether it's improve plantation husbandry.
Yeah, right. So, yeah, sorry. So, I mean, the seeds that you plant, the palms that you have is one thing, of course. The husbandry and the agronomic practices after that are something completely separate. And I think, as I mentioned at the end of my presentation, I think Dr. Hari will touch on it later, is the palms themselves are just one thing. It has to come as a whole package in terms of agronomic practice, in terms of mechanization, automation, to make sure that the potential of those palms is actually realized at the end of the day.
Right, from Nurul Iza Abdugani. So far, when you upgrade your breed through introducing new beneficial genes through crossbreed, have you encountered any negative impact of heterosis?
Negative impact? No, so far, I think we don't see anything yet. Yeah.
Right, the Ying Sing is following up on the question earlier. Can we clarify how, if there is insufficient data available to conclude the results of the yield performance, but SD is planting genome select seeds in the large-scale manner? Can I read that again? Appreciate to clarify how, if there is insufficient data available to conclude the results of the yield performance, but SD is planting the genome select seeds in the large scale.
Okay, I think the question earlier was related more to a scientific publication where we would want to have at least 4 years of commercial data before we were to have a scientific publication. So internally, we've been monitoring the yields for coming up 3 years now. We've been looking at oil content analysis on a monthly basis of those palms in more than in 2 locations. And so far, as I said, both yield and oil content is consistently better. And we're very encouraged by that. We mapped this yield against all the other fields that are planted at the same time and continue to be encouraged.
I think maybe I can add, just to clarify things. The GS model was developed on an existing population of palms that have full data set for over 10 years, right? So that prediction was basically applied on that population. Then we look at the real data of what we achieve, and then we have a certain accuracy value, right, that was published. So it's 0.57, 0.6, R squared of 0.6, right? So, and that includes the vagaries of climate as well in that, that total outcome, right, of 0.6. So we're quite confident, given that we are selecting materials from that pool, that they will perform well. So the first few years of data that we have at the moment basically just verifies it.
All right, thank you. While we're on the scientific line of questioning, there's another question here that I hope I get this word right. Is it possible through GS to choose an oil palm parent that has high homozygosity? And is it possible to assemble D by P progeny with high hybrid vigor from these materials?
Yeah, I think this is a good question scientifically. So I think this one approach here is to create some sort of F1 generations. So first you try to do some self-pollination, a few generations of the mother side, the dura. So using the marker as a compass, then you know in which generation you are reaching, like maybe it's like up to 80 or 60% homozygosity. Then after that, you will try to cross with one same father or something like that. So in that way, if let's say the traits is OU also at the satisfactory level, then that definitely can narrow down the standard deviation of your D by P population later. Yeah, it's possible.
Right, thank you, Chicken. Now going to another question. This one is a bit more operational in nature. When we double our planting yield and fully replant with GS, would our mills be able to operate at full capacity on internal FFB supply only?
Maybe I could take that one. So our materials that we are now planting basically are selected for what we call climate resilience in open and closed inverted commas. It also means that the crop produced is more even throughout the year. So normally oil palm traditionally production will follow two peaks, right? With a trough in between. And that means that the mills will be most probably running at over 100% capacity at certain months of the year and then under capacity at other months of the year. But with these materials, given the selection that we've deployed, we hope to even that out. So proof of the pudding is in the eating. So we will see over the next few years whether that remains true or not, and how big the deviation is between the high peak and the trough.
All right. From Mr. H. Go, Miss, how is this technology transferable to subsistence farmers? How long before the older generation of oil palm can be replaced by new planting materials in Malaysia?
Well, it's transferable in the fact that the seeds that we produce, we can sell to external parties, including smallholders. So the genetic material that we have is available for planting and will be available for planting, at least for Genome Select, in 2 years' time for sale outside. And as I said already, we sell seeds, and we sell seeds that are called Calyx Q6, which have got guaranteed genetic purity and traceability that are available for others to plant. So that is the easiest way to transfer this technology.
Right, yes, that answers two questions here, which was basically, how can we buy GS? And I think you've answered them when. That will be in 2 years' time, right? By 2023. So the how, I think they can ask us back in 2 years' time, right?
Well, yeah. So yeah. Sime Darby Plantation Seeds and Agricultural Services sell Calyx Q6 as well as Calyx 600 today.
Yes. These are seeds readily available using the same technology that we've been using to develop.
So Calyx Q6 uses the same genotyping platform to select within our previous best plant materials to have ones which are genetically pure, and we can show that and prove that, and they can therefore be tracked through to the field. Each seed has a basically a code on it, and this is something that is available to the public now.
Thank you. Thank you. Okay, we have one question. I believe this may be from academia. Basically, they're thanking us for the great talk, by the way, and they want to know if we have any technology transfer or training program provided for the public on our Genome Select research or Genome Select program.
Haven't thought about it yet, But since the suggestion has been made, most probably we can think up something and maybe offer it to the universities, right? So what is not so-called trade secret to us, I think we're quite happy to share, right?
Yes, I think as we can see from the talks that we had today, Sime Darby Plantation very open to partnerships, you know, at all levels for that matter, and we would like to share what we have learned with the rest of the world, or even the rest of Malaysia for that matter.
I mean, I think it's worth adding that, you know, we've published a lot of this along the way, and our lab has a steady flow of interns from the universities around us who come and spend time in our lab and help us out. And as Dr. Hari mentioned, you know, they learn a lot of the tools of the trade in this time.
Right, before we go on to the closing, just one more question from Mr. Eddie Supriyanto. He says, as you mentioned, you planted GS seedlings on the 180 palm per hectare on the coastal and 160 on inland. Any reason behind the choice of those densities? Is there a thinning out plan to reduce the density?
I guess that was for me.
You can do that.
Sime Darby Plantation has this policy, particular density policy, right? So most of the coastal areas are flat and therefore allows for higher density plantings of 180. And we've done trials for 20, 30 years on this. And there is a plan to thin it out. It normally happens after somewhere between 9 to 11 or 12 years when they start thinning, and a certain percentage of thinning that is done. And based on this, we know from previous trials that the actual total economic product or benefit over 20-year period is higher than if you planted it with a lower density. In the inland areas are slightly different because the soil types are poorer and so on and so forth. And also quite often not flat, it's sort of undulating slightly. What we found from our trials of planting 180, 160, 148, 136, and so on and so forth, is that the optimum is 160. So that's a simple answer.
All right, thank you, Dr. Hari. All right, ladies and gentlemen, any questions from those here in the auditorium, by the way? Or have you put them in the chat? Stuff like that. I don't see any hands here. No? Right, if there are no further questions here, we hope that we've been able to answer all the questions that have come in to your satisfaction. I think without further ado, I'd like to invite Dr. K. Harikrishna, the Chief R&D Officer of Sime Darby Plantation, to close our session today and give us a glimpse of how he sees the way forward in future-proofing the palm oil industry. Dr. Hari, over to you.
Thank you very much, Alfian. Good morning and good afternoon or good evening, wherever you are around the world. I have the first slide, please. Well, as you heard, we have massive issues to deal with. We all know about the availability of land or lack of availability of land. Definitely now with COVID, we don't have labor either. There are massive challenges of climate change, also consumer demands. A lot of questions about Ganoderma, so disease as well. And our competitors are not sitting still. You know, they are innovating and so on and so forth. And as Dr. Appleton mentioned earlier, our ultimate goal is targeted increases in both what we call potential yield, but actual realized plantation yield, estate yield. That is what really matters, right? What we get in research, yeah, gets us researchers excited, but doesn't get the corporate people very excited until we can deliver that value in the field. Can I have the next slide, please? So essentially this, you know, I always look at corn because corn is very simple, very similar in terms of the breeding process and the genetics compared to oil palm, right? And you can see on one axis, it's the number of bushels per hectare. And the other axis is the number of years, right? So you can see in the past when farmers collected seed and planted, replanted, and so on and so forth, the yields were pretty poor until most probably around 1935, 1936, whatever it is, when more advanced scientific methods were deployed. And you can then see this dramatic improvement in yields, right? when they started doing so-called double crossing, and then to single crosses, which are essentially making inbred lines to cross them. And today, with advanced biotech approaches of using GMOs, and then now gene editing and so on and so forth, you can see that line has increased further, right? So this is basically the kind of advances that modern science, deployment of modern science can actually deliver. And this is what our competitors are targeting and delivering. So unless we can actually start moving our improvement lines at a similar rate or better, we've become a dinosaur. Next, please. So, as I mentioned earlier, there's massive challenges that we have to face. And if you look across the whole value chain of oil palm, right, if we want to deliver high amounts of high quality, traceable, sustainable oil without increasing our carbon footprint, our water footprint, and being more sustainable than what we are today, then we have to have innovation across that whole value chain. So my colleagues talked about the genomics part, which I won't talk so much about, but you know, precision agriculture has a great deal of potential to play in terms of helping us to increase our productivity. And essentially, it's using advanced imaging technology from satellites, from drones, sensors on the ground, Internet of Things sensors, and digitization to actually put all of this together to make us more efficient. Then we're talking about labor issues. So mechanization and more advanced than that, robotics, right, will allow us to actually develop autonomous harvesting machinery, and we really need to have innovations in this space. When we come to the mill designs, basically the design goes back to the 1950s, and not a huge amount of innovation or changes have been made, despite the fact that technology is advanced so far. So in this example here, I talk about using enzymes to increase extraction rates, but there are a whole load of other technologies that can be deployed in the mills to improve productivity. Then you come to the refinery, and we need to actually improve the productivity of the refinery. And by utilizing IoT, deep learning, data analytics approaches, you most probably could actually optimize the refinery process as well. Can I have the next slide, please? So this is basically in a bit more detail about precision agriculture. So we try and combine data from various sensors, whether they're optical sensors on drones or satellites, and then rainfall gauges and so on and so forth, into a mobile app that the estate manager or assistant can use. And basically, we apply advanced mathematical tools to make sense of all this data that comes from various sources to provide us advanced insights where we can predict potentially when we're going to have maybe a disease outbreak, let's say in this case of bagworm, or it could be of another disease, right? So this is basically where we're heading towards by using some of these precision tools. Next, please. Now, as I said, we require continuous innovation across the whole value chain, and that is not possible for a single individual, neither is it possible for a single organization to do all of this. And therefore, we need collaboration with all of you participants, you know, as well as those who are in other parts of the world, perhaps not participating in this seminar today, but we need to actually have collaborations across into different technology platforms that perhaps will provide us solutions for some of these problems that we face today. Next, please. So as we start deploying more innovations, so on the furthest left of your image, you will see a field there. That is our first 50-hectare planting in Sungai Buaya, right? It's the coastal planting, and it's a drone image. So we've used these drone images to actually measure growth rate, right? So instead of measuring individual palms, there are about more than 9,000 palms there. It's not possible to do it physically. But by using these kind of imaging technology, you're able to increase the amount of work that can be done in a short space of time, right? And then we can overlay that on the soil type. And then you can see whether the soil is actually influencing the genetics or not. Is it really the genetics coming through, or is it the environment that is causing that improvement in growth rate? The images below basically is a LiDAR image of palms, so you can measure above ground carbon. We have a CAT scan computer, an X-ray image of a bunch. So basically, we want to optimize the carbon partitioning. As you can see, the stalk is very thick. And if we can reduce the amount of carbon that goes into the stalk and push more of that out into the fruits, then hypothetically, you can increase yield. So the question is, how do you do it? So this is what these young guys here are meant to do. Then we have all these mobile tools that allow our team in the field to actually monitor various aspects. And on the image on the right with a lot of green squares on it, this is an image of the lower side of a leaf. And we have an AI that actually counts the number of stomata there. So it allows us to identify basically potentially drought-tolerant types. Next, please. So the ultimate aim, as Dr. Appleton mentioned earlier, is to double yield, right? So that goal is 8 to 9 metric tons per hectare of oil, right, across all environments that we plant in by 2050. So that is our challenge. And if we are able to do so, we will actually significantly reduce that food gap, that 56% food gap that we are looking at the moment when the world population hits 10 billion. And that requires basically all these various platform technologies to be integrated together, from genomics, agronomy, microbiology, precision tools, as well as robotics, mechanization, improved milling, and improved refining, right, and digital. All of this needs to come together to actually realize that 8 to 9, 10 metric ton oil target. Next, please. I think that's my 2 cents worth.
Thank you very much, Dr. Hari. And of course, thank you to all our panelists for being here today. And thank you, Dr. Gwilliam, for joining us all the way from the UK. Ladies and gentlemen, harnessing the potential of increasing yield to sustainably meet demand is not the endeavor of any one business, or even all the major producers for that matter. It will require all stakeholders from across the entire palm oil industry. It is time for us to close, and I'd like to thank all of you very much indeed for your participation. Thank you for sharing your perspectives, and I hope you have found it a thought-provoking hour and a half. So goodbye and stay safe, everyone.